abroad. So I'll continue. Some of you may have heard Valery Krzyżanowski's lecture on Monday already introduced the topic of cellular senescence and its roles in aging. And this is a very interesting and dynamic field that, starting from the discovery of Hayflick, of the gradual aging of senescence of normal cells in culture, which for about 40 years stayed as potentially just a tissue culture artifact, and in the last period really exploded in terms of the discoveries of the roles of senescence. And you can see here that it has the privilege of being part of both of the hallmarks of cancer and hallmarks of aging diagrams. And it is indeed a very fascinating link between the two processes. Senescence is basically a cell damage response, one of the main mechanisms that cells respond to either DNA damage, activation of an oncogene, but also it's programmed in some situations to be activated during development and during aging, which is a very interesting phenomenon and field. And almost everything about a cell is changed once they enter senescence. The hallmark is that they will stop dividing, probably permanently, but their morphology dramatically changes, metabolism changes, and often these cells adopt a secretory phenotype. This was discovered about 20 years ago by the late Judy Campisi, and really changed the field in terms of viewing the senescent cells from a cell-autonomous entity into an entity that interacts with the immune system, with the tissue around it, and exposed the potential for these cells to do much more than was done. And what we've realized gradually over the last 10-15 years is that as we age, almost every tissue in our body accumulates cells that are in a state of senescence, and almost virtually any type of aging-associated diseases, many groups have been able to find senescent cells appearing in the course of the disease. Here's a very partial list. Essentially, these days, I feel that almost any disease, especially if it's associated with aging, if you look for it, people will find senescent cells. And a dramatic breakthrough from this work by Jan van Deursen from 8 years ago, using a genetically manipulated mouse trick. They were the first to show that these cells not only accumulate with age, but if we remove them as a mouse, in this case, ages, it really has a dramatic effect, an extension of lifespan among the largest you can see in a model animal, 27% median, and health benefits, so delay of aging. So these cells are accumulating in our body, they're contributing to disease, they're killing us in a sense, so it would be a good idea to get rid of them. And this really opened the door to the field of senolytics. Can we find a drug that actually can kill a senescent cell specifically and remove it from our body? Here's one of the early examples from the work of Peter de Keyser using a peptide, a FOXO4, where not only, here's in the aged mice, here's the treated mice, not only delayed aging, but even reversed part of it, which is very impressive. And that opened the door to a flood, a huge effort over many groups and companies in the last few years to develop drugs that will either kill senescent cells specifically or block their negative actions, inhibitors of BCL2, anti-apoptotic proteins, and many others that are not all listed here. Together with Valerie, who spoke on Monday, actually in this study, this joint study together, we were among the very first papers to show that it is actually possible to remove senescent cells in vivo. In this case, from a mouse model that we developed, Tony Tokarski and my lab developed, where we can generate senescent cells here in the epidermis. If you treat with this pan-BCL2 family inhibitor ABT737 just for a couple of days, you can get rid of about half or more of these cells. And in this case, we can even stimulate the proliferation of hair follicle stem cells in the tissue. So we can remove them, and this may have a beneficial effect. And as I said, there's many senolytics that are being developed, searched for, suggested, and effects on lifespan and aging. You can see various studies starting in 2019, and very recent dramatic publications that are just coming out, suggesting that different approaches to killing senescent cells may alleviate aging phenotypes and extend lifespan. And this is the remaining question, just from this month, Nature Medicine. It's very nice that you're doing all of this in mice, but can you actually do this in humans? And here's just a list of senolytics from 4 years ago, non-exhaustive, and 2 years ago, different types of clinical trials ongoing, mostly for aging-associated indications rather than aging itself. But we're waiting to see if really senolytics targeting of senescent cells may affect aging. So I'll try to tell you in the brief time two pieces of our own work. The first has to do with senescence in cancer. And as senescent cells accumulate in our body, that can affect cancer, but also as cancers develop, senescence is part of the process, mainly first as the most important tumor suppressive mechanisms that we have in the body. When a cell is damaged, has mutations, or oncogene activated, senescence is there to block it from multiplying. And that's the fundamental function. However, we see that as cancer develops, we find senescent cells within early lesions, advanced lesions, and it turns out they can do various effects. Some of them also blocking cancer, but others can advance cancer. And that raises the question, when and where do we see senescence in cancer? What are these cells doing there? And can we use senolytics and other approaches to induce or eliminate senescent cells to improve existing therapies? We've been interested in pancreatic cancer now for several years, also together with Yuval Dov, Gidi Zamir, Valery Krzyzanowski. And it's when we stain for P16, one of the main markers for senescence, either human pre-malignant or malignant tumors, we find senescent cells, P16 positive cells, also in mouse models of the disease. As you can see here, some of the senescent cells are within the tumor parenchyma, the epithelial cells. And in work done with all of the partners I mentioned before, some years ago we showed that these cells are actually pro-inflammatory in the pre-malignant stage. So actually they're supporting the progression from the pan to the malignant stage through generating inflammation. And therefore, if we get rid of them, we can really affect this transition in a mouse model. However, you can see that we also have stromal cells that are P16 positive. And as we advance to malignant disease, they're much more prominent. This is because as the tumor becomes malignant, it loses some of this basic senescence machinery, i.e. P16 itself, P53. However, it's been shown by other groups that when these tumors are treated, some treatments will actually induce senescence back in the tumor itself, and that can be leveraged for therapy. But we are focusing in this study on these cells that are actually turned out to be cancer-associated fibroblasts. So we learned through various stains and analysis that there's a subset of the fibroblasts in the tumor microenvironment that can become senescents, regardless of their identity and subtype. So they'll stop dividing. And then isolating them, growing them in culture, different methods, including isolation directly from patients that we can do by fax. This was a heroic effort by Benjamin Assoulin. Also analyzing published data sets. We can look at CAF, single-cell data sets, identify senescent cells. And all this leads to looking at gene expression profiles that indicate that the main thing that these cells might do is affect the immune response of the immune microenvironment in the tumor because of multiple associated genes. Actually, if you look at human tumors, you'll find that there's a negative correlation between the presence of senescent fibroblasts, shown here in red, where there's high P16 CAF, and there's few activated CD8 T cells in this environment, whereas without the CAF, there's more of these activated T cells, as you can see in these curves. And in fact, using mouse models of transplanted tumors, we found that if we transplant tumor cells that themselves cannot undergo senescence, we'll find senescence, cancer-associated fibroblasts in the stroma. And we can get rid of them, either using the genetic system that I mentioned before, developed by Jan van Dusen, or pharmaceutically using an a BCL2 inhibitor, in this case ABT199. And this is an FDA-approved drug used in leukemias and lymphomas called venetoclax. So if you use this, we can actually get rid of most of the senescent cells in the environment. And what happens, it's not enough to affect tumor growth. However, we see activation of these CD8 T cells, which are the main player in attacking growing tumors. So basically we understood that these CAFs here act to suppress the activity of the CD8 T cells. This led us to this experiment shown here, done by this really dedicated group of students, Benjamin Asulin, Rachel Khan, and Lutfi Odali, recently published, where we asked if we remove the senescent cells using venetoclax and we get a higher fraction of activated CD8 T cells, will that potentiate the effects of immune therapy? So here we can see that the senolytic itself doesn't do much. In this case, the immune therapy is quite effective, but if we add the senolytics, we actually eliminate most of the tumor cells. Whatever is left is necrotic. And there's a significant effect that we now expanded. Even if we reduce the effect that we only use anti-PD-1 and do it for a shorter time, the senolytics really synergize with this. And this altogether suggests that attacking senescent cells in tumor stromas might potentiate immune attack. And there's similar studies by others in pancreatic cancer and prostate cancer, lung cancer, targeting either fibroblasts or macrophages that in all of these cases seem to restrict the attack of the immune system. So this is a field that we believe that senolytics might be effective. If we stay in the same organ, the pancreas, and with some of the same partners, Yuval Dor, and we look at another organ that is present in the pancreas, it's the islets of Langerhans, which harbor the beta cells, the ones that are responsible to secrete insulin in situations where there are high glucose. And our interest in this allowed us to ask a general question. If we have a cell that we know has a very specific function, and as we age, some of these cells senesce, does it affect its function? And here we have a very clear and measurable function. Glucose, cell senses them, beta cells, secretes insulin proportionally. And if we look at this organ, as I mentioned and Valerie mentioned, various organs accumulate senescent cells. But these beta cells really stand out. As you can see with the stain of P16, as we age, this is one of the tissues where actually the fraction of cells that express senescence markers is among the highest. Here's another stain of P16 in an islet. You can see differences in prevalence. Almost 20% of beta cells in adults express this senescence marker. Here's RNA that shows the same. We can identify single cell analysis, also increase in age. It's a very striking percentage, which stands out among tissues. So that leads you to ask, well, are they really senescent, or is this an epiphenomenon, expression of some markers? And if so, can they still do this basic function? And we've been addressing this in several years. Here's a recent work from postdoc Milan Patra, and he started from analyzing these single cell data sets and finding the senescent beta cells and asking what are their molecular features. And there's three main features. One of them, we see general senescent signatures, other genes known to be associated with senescence. So we believe they're really in a senescent state. It's not an epiphenomenon. The other is that rather than displaying molecular features of dysfunction, they express features of functional maturation, suggesting that they haven't lost their ability to secrete insulin. In fact, they seem to be more functionally mature. And the last interesting feature was that they appear to have an activated interferon pathway that includes elevated expression of markers of the HLA antigen presentation system. And in fact, looking at both of these features, we see a very interesting phenomenon. On the one hand, in this case, the senescent cell seems to be doing something good. So if we do a culture trick, we take beta cells, we induce them to senesce, stop dividing. Actually, now they start secreting glucose and are more secreting insulin and are more responsive to glucose level. In a study from several years ago by Ronnie Hellman, we also showed this in vivo. If we induce senescence actively in islets here in red, actually this improves their insulin secretion and even can reverse features of diabetes. So here's a surprising effect which is positive. Actually, since then, people have shown in various organs and cell types that in some cases, senescent cells, as we age, this reflects their increased functional maturation. This has now been shown in several cell types. So that's very interesting. So that's the good part. Maybe it's not so good to target them in this case. However, a more suspicious part that is new in this paper is the discovery that I mentioned of the increased HLA presentation Increased response. Here's human islets isolated from cadavers, measuring HLA, and after stimulation with interferon, they respond more and they get to a higher level of presentation. We see this in general. So they're more antigen presenting. Why is that? One clue is that we can find elevated levels of cytoplasmic DNA and micronuclei in the senescent cells, shown here in green. This is cultured cells. Also in, oops, we lost some of the colors here, I'm sorry, but you can see this even in the senescent P16 positive cells in sections. So for some reason that we're trying to understand, that in vivo beta cells appear to accumulate double-stranded cytoplasmic DNA. And this apparently stimulates the basal level of interferon pathway, resulting in increased responsiveness and apparently increased antigen presentation. This, of course, doesn't sound so good as we age. And in fact, it's a big discussion in the field by others and us whether this phenomenon and other phenomenon can play a role in type 1 diabetes, when the cells are eliminated by the immune system, or also in type 2 diabetes. And there's various discussion really how good or how bad these senescent beta cells are. So to summarize, senescent cells, an important part of aging. In cancer, there really seem to be a developing and attractive target for senolytic drugs. In aging, huge activity in the field. We'll have to wait and see. Specifically, the role of beta cells illustrates, I think, the complexity of the functionality of these cells. And I'll thank my collaborators. Masashi Narita was my partner in the BIRACS, and my students shown here. Thank you very much.
Is there a burning question? Yes, I know one question.
This is a good question. I'm just curious about
microphone, please.
I will repeat the question.
I'm just curious if there's about lineages that are like long-lived, non-replicative cells. When you look at the results of inhibition of BCL2 pathway and the effects of like the aging phenotypes that you see, what happens in lineages like neurons which don't turn over? And do you see any?
So interestingly, some of the populations where we see senescence, including beta cells, are actually cells that are not replicating much during our lifetime. There's now really a focus on the brain. Just saw the paper by Fred Gage talking about neuronal senescence in aging, microglia senescence in Alzheimer's, glia senescence in Alzheimer's, but and in lab animals you can achieve the results with senolytics, positive results, but I think we still cannot answer the question I think that you're asking. What will be the global effect if we treat humans? So that's going to take a while, I imagine, to understand.